Hello, my name is Makaya Chilekwa, and I'm a rising senior at Cornell University studying chemical engineering. This summer, I worked with Professor Hadley Sykes from MIT on a research project that sought to design faster and more sensitive COVID-19 testing kits. My duty in particular was to develop a mathematical model that would help achieve this goal. So as we all know, in December 2019, a novel coronavirus emerged from Wuhan, China, and caused a severe respiratory illness known as COVID-19, and that resulted in a global outbreak. The absence of a working vaccine was a problem because the virus spread rapidly, and so to facilitate containment efforts, the testing time needed to be shortened in order to test as many people as possible. But then with the traditional detection method that typically takes 2 to 3 hours, one might ask, how do we reduce the testing time? So before we dive into that, a little bit about the coronavirus. So the coronavirus is an RNA virus and is very similar to the virus that causes influenza. The diagnostic testing approach that we were developing in the lab would work to detect the nucleoprotein or the N protein in saliva using a technique known as ELISA. So ELISA is a paper-based enzyme-linked immunosorbent assay and is preferred for its simplicity and expedience, especially with point-of-care tests. So how does ELISA work? The general idea is that you have the N protein that is sandwiched between two binder proteins. So you have the capture reagent that also binds to the test paper, as well as the detection reagent that would give the signal or the color change in this case for a positive test sample. So in our COVID-19 immunoassays, this is our desired complex that we need to form and optimize. So the testing protocol would involve mixing all the reagents into a microtube, and I was especially interested in simulating the reactions that would take place in the microtube under the assumptions that the mixing was ideal and perfect, and that the binding occurs on two separate sites of the N protein, and also that all the reactions that would take place are first-order reversible reactions. So the first step was thinking through the reaction chemistry. Because of non-specific binding, there are about 124 possible reactions that would take place when all the reagents are mixed in the microtube, and I included 4 of them, as you can see in the slide. The next step was actually building the mathematical model. So I developed a function that solves the rate equation in MATLAB, as well as a script that would generate the graphs that would help analyzing the different compounds that would form. And I included snapshots of that in the slide as well. So I generated a graph of the desired complex, and as you can see, the concentration increases for the first few seconds, but then decreases over time. So from this, we can see that longer incubation times would actually decrease the concentration, and only an incubation time of 160 seconds would give a very strong signal. I went a step further and varied the initial concentrations of the N protein, as well as one of the binder proteins, in this case, the labeled binder protein. And as we can see from the graph, decreasing or varying the concentration of the N protein does not change the results much, but decreasing the concentration of the labeled binder protein increases the fraction of the desired complex. So from this mathematical model, we can see that long incubation times would decrease the concentration of the desired complex, and shorter incubation times would be preferred. The next steps, however, would be optimization steps, in which we need to know exactly what concentrations would improve the fraction, as well as give a very short assay time to achieve the goal that we set out to achieve, and also refining and reducing the model by deleting non-essential reactions and just considering the kinetics of the reactions, because some reactions are more important than others. And that was my work. Thank you.